Okay, so I can't start my video because I have my document camera plugged in, but what we are going to cover today is the chemistry that is necessary for your biology class. So you have these papers posted for you under the chemistry of life. You have a blank copy like I have if you had a chance to print it out so you can follow along. There's also a completely filled out copy for those of you who don't have a chance to print it out and follow along. Either way, we are going to go through this worksheet. So, chem that you need for bio. First thing first, this is an atom. You know why you can't trust atoms? Because they make up everything. It's funny, you gotta laugh. You gotta laugh. The atom, as you see here, I'm gonna get some colors out. Everybody loves colors, right? The atom is made up of negatively charged electrons, which I am apparently coloring green, which you can't really see, but that's fine. The electrons stay out around this energy shell. These energy shells, actually, because there's one here and two here. The outermost energy shell is called the valence shell. And so those are called valence electrons. So this little doodad right here is a valence electron. Then you have neutrons, which I am apparently coloring purple, red, purple. You see here, the reason they are called neutrons is they are neutral. They do not have a charge. They do have mass though, and they weigh nearly the same as a proton, and the protons here are positively charged. So the mass of an atom is the positive and neutral charges of the atom added together. The electrons are so small, they do not add any mass to it. So let's write some information alongside our atom here for you to know about. Oh, let's see. This is fine. I don't know what color this is. First, this little bullet point here, atoms are made up, atoms are made up of subatomic particles. So an atom is going to be a proton, a neutron, and an electron at minimum. Those three things will form your basic atom, which is hydrogen. And then it's just information that I've already talked about while we were coloring our atom here. Electrons are, I'm going to just do this, it means negatively charged, and are found in shells. We can, air quote the shells, or it's also known as an electron cloud, because in reality, they are not nicely isolated into these shells. In reality, because at no given time can you ever say exactly where an electron is, they just call it an electron cloud. And this is outside the nucleus. And my handwriting got a bit too big, so I'm a little off my little chart here. Protons and neutrons, we know, are found in the nucleus. Right? And then the final bullet point is that protons are positively charged. I'm just going to put a positive. And the number of protons determines the element or the type of atom you are looking at. Number, that's number. number of protons determines, I'm just gonna write that across, determines the element. We call it an element, in which it can also be called the type of atom. And so you know the periodic table. Of course you know the periodic table. The periodic table is all of the atoms / elements, whichever word you want to use, that we know that exists in nature or not in nature. We have some man-made ones is what that means. And they are organized neatly based on electronegativity and mass size and how they interact with each other. So that's what the periodic table does, and we'll talk about that later. But everything, including living things, are made up of atoms. Hence my joke, why can't you trust an atom? Does it make up everything? Okay. That's what makes up an atom. Isotopes are atoms of the same element that have a different number of neutrons. So let's look here. We have our element, and I'm actually going to take the time really quick to squiggle in my electrons as green, which once again isn't showing up really well, but it's just, you know, to give some visual interest. But the inside, you see, is not separated into positive and negative charges. The inside just tells me we have 6 protons and 6 neutrons, and then we have 6 protons and 8 neutrons. Remember, the neutrons determine, the protons plus the neutrons determine mass. So this is a higher weight atom, even though they're both carbon. They're both carbon, 6p, 6 protons. And so these are called isotopes. So let's write some information down. So these have the same number, and I'll write out number, of protons. These have, and I'm going to make this big, a different number of neutrons, thus making it an isotope. And that's, okay, well then, and they are called isotopes, which also is the name of the baseball team in The Simpsons. Yeah, fun fact. Why do we care about isotopes? They're radioactive. Radioactive. And so biologists, write that down, biologists use isotopes in research. Specifically in terms of carbon here, for carbon dating. Have y'all heard of carbon dating before? It is a way to determine how old something is based on the half-life of the carbon-14, because 6 + 8 is 14, isotope. So radioactive elements have half-lives, which is how long it takes until only half of it remains, basically. And so you can use the half-life of carbon for carbon dating. Fun fact, I don't know if you know this, archaeologists say you can determine the difference between rock and, what's the other one? Bone, thank you, by touching it to your tongue. Rock doesn't stick to your tongue, but bone is porous, and so it'll stick to the tongue. So you have a bunch of archaeologists running around licking things to try to figure out if they're rock or bone. I don't know if you needed to know that, but now you do. Radioactive isotopes can also be used. It's hard to talk and write because you're trying to use 2 different language centers in your brain. Can be used to label molecules in experiments. And if you have room to write in experiments, then go ahead and jot that down. And we're going to see that a lot more when we talk about in our 4th. So you have chemistry of life, and then you have the cell, and then you have metabolism of the cell and enzymatic activity, and then you have DNA and transcription and translation. And you're going to see a lot about how we use radioisotopes in history to actually figure out that DNA is the genetic material and how it functions. Long time, they were arguing, was it DNA or proteins that was the genetic material. So that is the function of isotopes as we use them. Also, you know, we have bad isotopes like plutonium. If we were ever to have a nuclear war and nuclear fallout, those would be bad things for us. So down here, and it's lighter for some reason, but it says ions are atoms that have more or fewer electrons than protons. That's why they are charged. So remember, we have electrons, protons, and neutrons. This is telling us what happens when the neutrons are different than the protons. We get isotopes. Ions are what happens when the electrons are different than the protons. And these are charged, right? Protons are positive, electrons are negative. So if we have a difference in positivity and negativity, you're going to get a positive or negative charge. So that's how ions work. And ions are a big way in how atoms interact with each other to form compounds and molecules. So we have a sodium atom here, 11 protons and 12 neutrons. And sometimes naturally the neutrons are just one more than, like up here in our example, you had 4 neutrons and 3. That doesn't throw off the charge, and that's also not an isotope. That's just the way they are naturally. So a normal sodium ion has 11 protons, 12 neutrons, but 11 electrons because the charges have to be equal. We can take electrons away, and in fact, sodium wants to get rid of its electron. And this is about filling the valence shell. This valence shell is only full if it has 8 electrons on it. Sodium only has 1 hanging out there. It's very unstable. And so it's not easily going to get 7 more electrons. It would have to form 7 different bonds. But it can easily give up an electron. And so in a lot of compounds and molecules, you'll find that sodium gives up this electron to fulfill another valence shell of another atom. And then it's happy and the other one is happy as well. So here we have a sodium ion that is positively charged because we got rid of a negative charge. So now we have more positive charges. We have 11 protons than we do neutrons. So you have a positive charge. We could have added electrons, but you'd have to add, I don't know, you have 7 more to be stable. Let's say we added 2 electrons. And so you had 3 hanging out in this valence shell. So you had 13 electrons. Then that would be an Na2 minus. I don't want to write it down here, but then you would get Na2 minus because you'd have 2 more electrons. Or let's say we got rid of 3 electrons, then you'd have Na 1, 2, 3 plus, right? That makes sense. I'm crazy. And so that's how the charges work in ions. Okay, so sodium, highly reactive because it only has this 1 electron on the valence shell, usually reacts with things like chlorine and fluorine because they have 7. And so they want that extra 8 things. And so when we get sodium with chlorine, sodium is an explosive soft metal. Chlorine is a poisonous gas. Add them together and what do you get? You get salt. Table salt to be precise. But usually when you add one of these reactive metals to one of those metalloids, you always get something we call a salt. Sodium fluoride is a salt. Magnesium fluoride is a salt. Those types of things. So it's a salt. So follow along here. When solid salts, and our example is like NaCl, dissolve in water, they form ions. Ions are charged, which means they can actually cause charge to flow through water. Positive ions have fewer electrons and negative ions have more electrons than normal atoms. In the human body, in biology, we have taken advantage of the fact that when salts dissolve, they form ions, and we use ions to communicate between ourselves, between ourselves and ourselves. That's communicate. Kate just got cut off there. Between cells, such as nerve cells. Such as, we use it for all cells, but nerve cells. Ions are also important because many biochemical reactions occur when there is only a particular salt concentration in the cell or solution. That also goes along with nerve cells. You have something called voltage channel gates in your nerve cells, and the voltage comes from ions. So you have to have a certain amount of potassium and a certain amount of sodium in your body to open and close those gates. So many biochemical reactions, and I'm going to shorten reactions to RxNS. It's not really shortened, but it's a form of shorthand that I created in college. I'm sure I didn't create it. I'm sure I watched a professor do it, but then mimicked it. And if you can learn how to shorthand for yourself while you take notes, in college it'll come very handy, because then you'll go back through your class notes and add in your textbook notes and write it all out fully, and you'll have a very comprehensive, but you have to keep up with the professor in college, and it works if you make different things for yourself. So anyway, reactions occur only when a specific, I'm going to say because it's a smaller word, specific salt. And here's another shorthand. I'm pretty sure that I saw this in a chemistry teacher, but this is for concentration. It's just open and closed brackets. So many biochemical reactions occur only when a specific salt concentration is in the cell or in solution. So you have to have a certain level. Our body cells, we are salt. We are made of salt water, actually. And so we have a normal concentration of salt similar to saline. Normal concentration, again, of salt in body we call saline. And so if you're in the medical field, or you plan on going in the medical field, when you have to give fluids to a human being, you can't give them pure water, because that is not what we are made of. You have to give them a saline solution, because we need a specific number of ions in our solution in order to function properly. Okay, so those are the types of atoms that we have. We have a regular atom, we have isotopes, and then we have ions. What do we do with atoms and isotopes and ions? Well, we don't really do anything with isotopes. You have isotopes in your body because they're in nature, and you eat them, and then it gets digested and embedded in your cells. But you're not like made of isotopes or anything. But we need to use these atoms to create molecules. We are made of molecules. We are made of cells, but we are made of molecules. Molecules came before cells were created. These atoms, using whatever they have in their valence shell make different types of bonds. You have covalent bonds, ionic bonds, hydrogen bonds, van der Waals bonds, sulfur dibridges, disulfide bridges, two sulfurs, whatever. You make all sorts of bonds. And so you have a hydrogen atom here and a nitrogen atom here, and since they're sharing that electron, they have made a covalent bond. Sharing is covalent. Taking is ionic. Chlorine over here has taken, remember, that single valence electron that sodium has. It has taken it. Now chlorine has 8 valence electrons. Sodium lost an electron shell, but it now has 8 valence electrons as well, which makes them both stable, which is good. But it took that electron. It didn't share it, so that is an ionic bond. And we'll talk about hydrogen bonds in a minute. And you'll find that covalent bonds, ionic bonds, and hydrogen bonds are the 3 most important bonds in the human body and in biological life processes anyway. Okay, over here in this little corner, we're gonna write atoms and molecules. Are held together by covalent bonds. And I wrote that really big. I don't know what I was creating. But it doesn't matter because all I have to write is that covalent bonds means we're sharing electrons. So covalent bonds equals sharing of electrons. And it's very important you understand the sharing. This is a shared electron pair, which means that the electron from hydrogen and that electron from one of the electrons on the valence shell of nitrogen, because you see it has more bonds over here, they're equally in between the hydrogen and the nitrogen, and they are both sharing the energy charge from that electron. So conversely, we have ions, right? Oh, sorry, covalent bonds equal sharing of electrons. Ions, or ionic solids, so salts, and I'm even going to put salts in parentheses here, are held together by ionic bonds. Okay? Ionic bonds, I've already said, is a taking of an electron. It is transferred to another atom, and that's the word we're actually going to use. Ionic bonds form when one or more electrons are transferred sorry, my handwriting's getting a little sloppy, transferred from one atom to another. I've always done this thing where I kind of do a half cursive, half printed form of handwriting. It's always how I've done it, so I don't know. When that ion is transferred from the sodium to the chlorine, for example, the sodium, one loses electrons, and so it becomes positively charged, you can see that, and one gains electrons, or an electron, so it becomes negatively charged. So the one that loses I'm just going to write this an E with a negative charge for electron, that's how I denote electrons, becomes positively, once again, just positive plus sign, charged. The one, and I'm going to run out of room here, that gains the electron, once again, E with the negative means electron, equals, or yeah, becomes negatively charged. Oh, I forgot my little negative sign. Oh well, wrote out negatively. Negatively charged. So these bonds are atoms. joining together. So molecules and ionic solids are made of atoms. Okay, some molecules act differently in water. And you have to remember, the whole point of this, we haven't talked about water yet, and you're gonna see just how important water is to life. But we are made up of water. We evolved in water. We were water animals before we ever emerged onto land, which means all of our life processes have to occur in water. So the fact that molecules act differently in water is one of the reasons we can do certain life processes in water. Okay, so some molecules, we're looking here. Some molecules are attracted to or stick to water. These are things that easily dissolve to water molecules. These are things that easily dissolve in water, like salts and sugar. Sugar is very big as a molecule, so it takes a long time to dissolve, and you usually have to heat it up. But they will dissolve. If this is true, that they like to stick to water, these are polar. We call them polar molecules, and they are polar and hydrophilic. So hydrophilic is a Latin term. This is an H. Hydro meaning water, philic meaning to love. So they love water. They're polar and they love water, okay? And these often have nitrogen, oxygen, or sulfur atoms. I'm just gonna squeeze sulfur in there, atoms. Okay, and so you're like, what are you talking about, Miss Millsmore? I just don't understand. I got you. I'm going to circle the water molecules in this picture, all right? This is a water molecule. Here's water. Here's water. Here. Here. You don't have to do this, but this is to show you where all the water is. Okay, so that is our molecule that we are submerging into water. And you see how closely the water has associated with our molecule. So when things dissolve in water, what actually happens is they have a hydration shell that just coats the thing, and it doesn't like disappear or anything. It's just completely coated in water. And so it is dissolved in the water. Conversely speaking, look here, I don't even have to circle them. Look how far apart these water molecules are from our molecule. Here, look, we have nitrogen, we have oxygens, we have oxygen over here as well. The water really wants to associate with these. If you see here, all we have in our molecule are carbons and hydrogens. And the problem here is that carbons and hydrogens, when they are fully bonded, so every time carbon has 4 bonds like this, and every time hydrogen has a singular bond, they're neutral. They're completely happy. They don't need to interact with anybody else because they are completely bonded and stable. These are facts. These ones, even though they have all the bonds, they don't have any negative or positive charges, which means we're not losing or gaining electrons here. Even though they have all their bonds, they still want more electrons. They're very greedy. The nitrogen wants more electrons and the oxygen wants more electrons. So it pulls hydrogen towards it. And that's called a polar bond. And we're going to talk about, not a polar bond, that's called a hydrogen bond. And we're going to talk about that in a minute. So easily dissolved in water and then not easily dissolved in water. So let's talk about these. I'm going to put down here, facts. Some molecules are not attracted to water. I'm going to attract it to water. And they stick together, I don't want to use the word stick again, and clump to each other far away from water. Right, and you see this when you drop oil in water. You've all seen a lava lamp. A lava lamp is just wax heated up in water. Well, wax is a fat. Wax doesn't want to dissolve in water. And so when it gets hot, it turns into a liquid. It rises to the top of the lava lamp because heat makes it rise, but then at the top it cools down, so it starts to sink again. And that's just how a lava lamp does, over and over and over. But that wax never mixes with that water. What do we call these? These are non-polar and hydrophobic. So here's Latin again. Water fearing. A phobia is to be afraid of something. So water fearing, water loving. And in fact, let me use, what color is going to show up really well? I guess just red. I'm going to write water fearing, water loving. That's what those terms mean. Okay. Couple more items. They are mostly made of carbon and hydrogen. Sorry. Okay. And have few oxygens or nitrogens. And when we get to macromolecules, which comes at the end of this chemistry of life unit, you'll see we have something called fatty acids. And fatty acids are tails, we call them tails of carbons and hydrogens, but they are bonded to 3 oxygens. And it's a backbone, a 3 oxygen backbone. And so they have few oxygen, but mostly carbon and hydrogen. Final point. The tendency for hydrophobic molecules to stick to each other and keep out of water, so all of this is called a hydrophobic interaction. It's what makes oil clump together and stay out of water, and wax and butter and all the other fats that don't like to work in water. So nonpolar hydrophobic molecules work differently in water than polar hydrophilic molecules. Our final point today, hydrogen bonds are really strong and really important. Okay, I'm actually going to take off the light for a minute. Nope, wrong button. Hydrogen bonds are really strong and really important. That might have been better the whole time, but I wasn't really sure when I started. So sorry for having the light on. Okay, hydrogen bonds. Water, specifically water, I'm gonna put another piece of paper under here for a minute. Let me talk about water for a minute, even though we're not on the water unit yet. You have an oxygen. Okay, so I'm gonna put oxy, and it is bonded to 2 hydrogens. Okay, sorry that they're different sizes, but that doesn't matter. The oxygen is happy to be bonded to 2 hydrogens. That fills its valence electron shell, but the oxygen is more powerful than the hydrogens. And so instead of having electrons Instead of having electrons equally shared between the 2, what happens is that the oxygen pulls the hydrogen's electrons towards it. So the electrons are pulled towards oxygen. Okay, and this is an electron, right? The electrons are pulled towards oxygen. What happens when that happens is, even though it is a covalent bond, the oxygen has more of the electrons around it, and it gets a partial negative charge. And the hydrogens get a partial positive charge. Now, it's not a figure 8. It's just a Latin symbol. It looks like that. Kind of looks like a spoony D, or a D that looks like a spoon. Either way. Okay. This is called a dipole. Yes. This is called a dipole, where you have two opposite poles, positive and negative. Still a covalent bond, not an ionic bond. The oxygen hasn't stolen the electrons from it, but it's got a partial negative charge. When that happens in water, that partial negative charge, I'm sorry, I keep writing it to, oh, sorry, this is an H. Let's do that. Partial positive, partial, oh my God, okay, positive. The other hydrogens that are closest to this oxygen, they're being pulled to the oxygen. So you'll have a hydrogen here with the water. So here's an oxygen, here's a hydrogen. And the oxygens in the water are being pulled to the hydrogens. And you have to think, in a glass of water, in an 8-ounce glass of water, are more hydrogen and oxygen atoms than there are sand on a beach. I'm not even lying. This is like a statistical thing. So all of those oxygens are being pulled towards the hydrogens, and they form this partial bond. It's not a real bond. One of them easily broken, and in fact, two of them easily broken. This bond here and this bond here. The problem comes when you have billions of them, and it's not a problem, it's actually a very good thing for us. When you have billions of these partial bonds in a glass of water, in the ocean, in a lake, they're nearly impossible to break. And it gives water all its amazing properties that we need in order to survive on this planet. So these hydrogen bonds are happening because of this partial negative and partial positive charge from the oxygen being super greedy and pulling the electrons to it, okay? So that's just all water. Here we're showing it with other polar molecules. So polar molecules are attracted to each other. Right? Okay. Hydrogens bonded to oxygen, like we see in the water, or nitrogen, are very attracted to nearby oxygens or nitrogens. Okay, these hydrogens become attracted to those other partially negative charged molecules, because remember, these are partially positive. And in fact, if I can just draw it without, okay, partially positive, and this is partially negative. And so that attraction occurs and you form a hydrogen bond. It's an R, A-R-Y, I mean A-R-E, R. Very attracted to nearby oxygens or nitrogens. This results in hydrogen bonding. Okay, and so that is it for your basic chemistry. I'm going to have a short quiz for you to do in a minute. Well, not in a minute, but after we are done with this segment, which is now, because this PowerPoint is over. This isn't a PowerPoint, this is a lecture. So let me stop sharing and